Okay, a warm welcome again, everybody, to the Kiel Imaging Seminar, and it's great to have you all here today. We have a fantastic speaker today, and I'm very happy to welcome Professor Jeanine Prompers. She's a professor of organ-specific metabolic imaging in the departments of human biology and imaging, NUTRIM Institute of Nutrition and Translation Research in Metabolism in Maastricht, the Netherlands. Her research focuses on the development of multinuclear MRS and MRI methods for studying in vivo tissue metabolism. Her research track record comprises the ultra-high field MRS to measure metabolism in skeletal muscle, liver, and heart. And she is among the first pioneers who developed and applied ultra-high field deuterium metabolic imaging of human brain and liver. Recently, she became CEO of Scannexus, the ultra-high field MRI facility in the Brightlands campus in Maastricht. And so before we start, to the audience, please wait with your questions until the discussion. And with this, Jeanine, the stage is yours.
Well, thank you very much for your kind introduction. I'm very happy to present, not live in Kiel, but online. So I hope to pay a visit to Kiel maybe sometime later. So what I would like to do today is, let's see if I can make this go away. Yes. I would like to show you our results on metabolic imaging, and especially how we try to translate this really to clinical applications. So since January this year, I actually moved to Maastricht, where I work at the university, but as was already explained, I'm also the director of Scannexus, which is the ultra-high field MRI facility in Maastricht, where we have 3 MRI scanners with field strengths of 3 Tesla, 7 Tesla, and 9.4 Tesla. And I also will use those for my own research. So to give you an idea of where we are, you are here in Kiel. This here is Maastricht, so in the very south of the Netherlands. But I'm not in Maastricht right now, I'm at home, and that is actually close to Eindhoven. And most of the work that I will show today that I performed before I moved to Maastricht. And then I was working in Utrecht, which is here, where I was a member of the ultra-high field MRI group. So I would like to start actually this presentation by thanking the people who did all the work that I'm going to show. So a couple of very bright PhD students from Utrecht, Lieke van den Wildenberg, Li Shen-ho, Narges Amadian, Mark Gosselink, and Hayhan Kurzan, and Dennis Klomp, who was the group leader at the time I was in Utrecht, and all the other members of the ultra-high field group in Utrecht. Okay, so metabolic imaging. Why would we do that? This slide shows the expenses on cancer medication in the Netherlands. I'm not sure if this is similar in Germany, but what we have witnessed during the past years is that expenses have gone up massively. And this is mainly due to increased expenses on new types of treatment, especially immunotherapy. And you can see that the expenses on immunotherapy have more than doubled in the past 5 years or so, whereas expenses on other sort of treatments have stayed more or less the same. Now, of course, immunotherapy is really revolutionary in the treatment of cancer, and it's very promising. However, what we also see is that these new treatments are only effective in a limited number of patients. So it's not uncommon that only 30% of the patients benefit from a certain treatment, and the other 70% does not. Now, because these treatments are so costly, and also because they have negative side effects, it's very important that we have methods by which we can predict the response, or actually the non-response to treatments in a very early stage. So that the treatment can potentially be stopped already early on to reduce costs, but also to prevent the negative side effects for patients that do not benefit from a treatment. So now imaging can play an important role here. This is an example from the literature, so it's not my own work. But this shows conventional MRI images of a patient with a prostate tumor. You see a T2 weighted image and a diffusion weighted image, and you can see that the tumor quite clearly. Do you see my mouse, by the way?
Yes.
Yes, okay, great,
yes.
Thank you. And on the bottom, you see the same images, but then taken 6 weeks after therapy, which was a combination of hormonal and chemotherapy in this case. And you see that there's actually hardly any change on those conventional MRI images. However, what they also did in this study, they performed a metabolic MRI scan. And with this, they measured the pyruvate to lactate metabolic flux. And you can see a very high metabolic activity in the tumor before therapy. And 6 weeks after therapy, this metabolic activity has reduced almost to zero, indicating that the treatment was successful. And indeed, after 6 months, so that's then the normal time point that the success of a treatment is assessed, it appeared that indeed the treatment was effective in this patient. So this really shows the potential of metabolic MRI. So let's dive a little bit deeper into metabolic pathways in tumors. So this is a very simple cartoon of 2 types of cells, a normal cell on the left and a cancer cell on the right. And you can immediately note the difference. And one of those differences is the increased uptake and usage of glucose in tumor cells. So they really use a lot of glucose, but then also the metabolic pathway of glucose is very different in a tumor tissue. So in a normal cell, most of the glucose is used in an oxidative manner to produce energy, because that's most efficient. That way you get most ATP out of a glucose molecule. But in a tumor cell, also in the presence of oxygen, actually a lot of glucose is transformed into lactate. And the reason for this, it's still debated, but probably one of the reasons is that this pathway, so the glucose to lactate metabolism, generates molecules that are needed for tumor progression, for tumor growth. So this, what is actually called the Warburg effect, so this conversion of glucose into lactate, can be detected by hyperpolarized carbon-13 MR spectroscopy, and that is actually what was done in this study that I showed on the previous slide. But recently also another technique was developed, namely deuterium MR spectroscopy, with which you can also assess the same pathway. And that is actually what I will focus on during my talk today. Another thing that is of interest is the high membrane turnover in tumor cells. Because they divide very rapidly, there's a high turnover of membrane phospholipids. And this can be detected by phosphorus MR spectroscopy. So what I would like to do today is give you a little bit of background on in vivo phosphorus and deuterium MRS. So what can you measure with it? Then talk a bit about the challenges and solutions of those for those challenges, for those techniques. And then I will turn to some applications of both phosphorus and deuterium MRSI with a special focus on clinical translation. So in vivo phosphorus MR spectroscopy is actually a really old technique. It was already applied in the 1980s. And already back then it was recognized that this technique gives a unique view into energy metabolism in tissues in a non-invasive manner. So there's really no other technique that can do that. And how can we look at energy metabolism? That's by detecting energy metabolites like ATP. So here you see the 3 signals from the 3 phosphate groups in ATP, and inorganic phosphate over here. So this is in the liver. In other tissues like skeletal muscle or cardiac muscle, you would also see a peak from phosphocreatine, which is the energy buffer in those tissue types. However, in the liver, there's no creatine kinase expressed, so there's also no phosphocreatine present. Another peak which is of interest is this signal here from NAD+ and NADH. And this is informative about the redox state of the tissue. Now, for the study of tumors, there are some other signals that are of interest, and those are the smaller signals in the spectrum in this area here. And those are signals from the phosphomonoesters, phosphoethanolamine and phosphocholine. Those are membrane precursors. And on the other side of the inorganic phosphate peak, the signals from the phosphodiesters, glycerophosphoroethanolamine and glycerophosphocholine. And those are membrane degradation products. And especially then the ratio of those PMEs to the PDEs, so the PME over PDE ratio, is informative about membrane turnover, which is then a marker of cell proliferation. So that's a very short introduction into phosphorus MR spectroscopy. So then, deuterium is a bit newer. Well, not really, as we will see later. But it's a method that is really in development at this current time. So what is deuterium? It's another isotope of hydrogen, and it's twice as heavy as the more known proton, because its nucleus has both a proton and a neutron. It's a stable isotope, and the natural abundance is really low, so it's 0.01%. So that's even 100 times lower than for carbon-13. It has a spin, of course, otherwise we couldn't do MR on this nucleus. But opposed to the protons and the phosphorus and carbon-13, it has a spin 1. So that means it's a quadrupole. And the gamma, so the giant magnetic ratio, is 6.5 times lower than for proton. That means sensitivity is lower. It also means that the frequency is lower. And at 7 Tesla, the frequency for deuterium is 45.7 megahertz. Now, this lower frequency also has an advantage at high field, because the proton frequency at 7 Tesla is 298 megahertz, and that gives all sorts of problems, because the wavelength is rather short, about 10 centimeters, and you have problems then with B1 homogeneity, etc., etc. And those problems we don't have for deuterium at 7 Tesla, because when you do deuterium spectroscopy at 7 Tesla, it's more or less the same as doing proton MRI at 1 Tesla. And that is quite easy to do. Now, deuterium MR is very hot and happening, but actually it's also not that new. It was already applied in the 1980s, like phosphorus. And this shows some examples from old papers. So this is an application of deuterium MRS in a mouse, where they administered heavy water, D2O. And right after administration, you see then a huge signal from the deuterated water, which then washes out again. But then they see a peak appearing from deuterated triglycerides, deuterated lipids, that have become labeled during the administration of the D2O. So this is informative about the new synthesis of lipids. And this is another very old example where they looked at bacteria cells. And in this case, they added deuterated glucose, deuterated at the 1 position. And initially, they observed then the 2 signals from the alpha and the beta glucose. But later on, there's another peak appearing, and that's a peak from alcohol, deuterated alcohol. Because these bacteria, they can transform glucose into alcohol, and that is what you observe here in these results. So then it went silent regarding deuterium MR spectroscopy, and that is mainly because the other modalities like phosphorus and carbon-13, yeah, they were more successful at that time. But about 6-7 years ago, the deuterium MR spectroscopy was sort of reinvented. So this started by a publication from the group of Wei Chen, where they infused deuterated glucose into rat. And in this case, the glucose was deuterated at the 6th position. So there's actually 2 deuterons per glucose molecule. And then they performed deuterium MR spectroscopy measurements of the brain. And before the infusion, you can already see a signal. That's from the deuterated water, which has a concentration of 10 millimolar at natural abundance, and that's easily detectable. But after the infusion of deuterated glucose, you see more signals. The signal from the deuterated glucose itself, and then a bit later you see some additional signals appear. So where does this come from? Well, if you look at the metabolic pathway of glucose, deuterated glucose in this case, and we see here the deuterated glucose, which is taken up into the brain cell in this example, which leads to the labeling of pyruvate. And then there's 2 pathways, as we have seen already before on the cartoon in the introduction. Pyruvate can be transformed into lactate, but the accumulation of deuterated lactate is very low in healthy brain. So you hardly see a signal from deuterated lactate in healthy brain tissue. And actually most of it will enter the mitochondria and go into the TCA cycle, where then glutamate and glutamate are produced. And they accumulate at sufficient concentrations that we can detect them. The resonance frequencies of glutamate and glutamine are quite close, so it's already difficult to distinguish them in a proton spectrum. But in a deuterium spectrum, the line widths are much broader than in a proton spectrum, so there we can definitely not distinguish the 2. So we see one signal for both, and we term that GLX. So that is the signal over here. That's the combined signal of deuterated glutamate and glutamine. Now, what is really nice about this, these sort of experiments is that it's really easily translatable to human studies. And that's because the humans can just drink the deuterated glucose solution, and that makes life a lot easier because regulations for oral administration are way less strict than for administration through an infusion. So this is actually what we have obtained in Utrecht. So it's a spectrum of a deuterium spectrum of the human brain after drinking deuterated glucose. And you can see that it looks very similar to the spectrum from the rat brain, the similar resonances. Now, why is deuterium MR spectroscopy so successful now? This has to do with the short relaxation times, and that's because deuterium is a quadrupole. So because it's a quadrupole, it has short T1 relaxation times. Here you see a figure from a paper from the Yale group. You see the T1s of different compounds at different field strengths. So for deuterated water, we are around to 350 milliseconds and not much change with increasing field strength. But for other compounds like glucose, the T1 is even way shorter, around 60 milliseconds. And that means that we can, in our experiments, we can do very rapid signal averaging, so we can keep the TR really short. And that's actually the main difference compared to carbon-13, where the T1s are really long. So you need a long TR. And this gives really the gain in sensitivity for deuterium as compared to carbon-13. Now, because the T1 relaxation times are short, of course, also the T2 relaxation times are short. This then leads to broad lines. But as you have seen on the previous slide, there's not that many signals in the spectrum anyway. So overlap is not that much of a problem. And it's actually also sort of an advantage that the intrinsic line widths are already large, because that means that you're relatively immune to B0 inhomogeneities. Or in other words, you don't have to put that much effort in shimming because the intrinsic line widths are already broad. Now, what is really a game changer in deuterium MR spectroscopy is that we can make this an imaging technique. So we can perform spectroscopic imaging rather than just spectroscopy. And this really makes a difference because radiologists, they don't like to look at MR spectra. So if we can provide them with images, then we have a much higher chance that it's going to be used in the clinic at some point. So the sequence we use for spectroscopic imaging is really simple, mostly. So it's a very simple FID MRSI pulse sequence, which is basically just a pulse and acquire sequence. So you start out with an RF pulse at a certain angle, and there's an acquisition, but in between, we put these phase encoding gradients. And we can apply phase encoding in 3 directions, so to really make a 3D metabolic image. And to make it fast or faster, we can use a short repetition time and low flip angle approach to shorten the scan time even more. And what is also nice is that for deuterium, and that also applies to phosphorus, you don't have huge water or lipid signals that you have to suppress, unlike for proton MR. So yeah, the sequence is actually very simple. You don't have to add any elements for suppression of other signals. So this idea was introduced by the Yale group, Henk de Vijter and Robin de Graaf, in their seminal paper in 2018. And so here you see how it works. So they termed this technique deuterium metabolic imaging. On the left, you see a rendering of the normal proton MRI. And then on top of that, you see the deuterium MRSI data. So you have, in all these voxels, you have a complete deuterium spectrum. Now, of course, you cannot really look at all those spectra. So how do you analyze such data? Or how do you get a feeling of what's going on? Now, for that, you can take the integral under a certain peak. So, for example, the area under the glucose peak, and plot that area with a color code on top of the image. So that's done here. So these are so-called metabolic maps. And then in one view, you can see where the glucose concentrations are low and where they are higher. And the same you can do for other metabolites like GLX. And this is then an example in a patient with a brain tumor. And in this case, they mapped the concentration ratio of lactate to glutamate and glutamine. And you can see a very high contrast in the tumor. And that's because of what we have seen before, the Warburg effect. So in the healthy brain tissue or the normal appearing brain tissue, there's very little accumulation of deuterated lactate and a high production of GLX. So the ratio is really small. Whereas in the tumor, it's the other way around. And this is really an advantage of DMI as compared to, for example, FDG PET imaging. Because with FDG PET, what you administer is that is compound FDG. And what happens with FDG is that after phosphorylation, it's trapped. It is not going to be metabolized any further. So you just look at the uptake of glucose, which can be, in certain tissues, can be high in both the normal tissue and in the tumor tissue, like in the brain, but also the liver, for example. And the healthy brain tissue also has a high glucose uptake in the tumor as well. However, the metabolic fate is pretty different for the glucose in the tumor and in the healthy brain. And because we can actually detect the downstream products with DMI, that makes that we can obtain a very high contrast between the tumor and the normal tissue. Okay, so that's phosphorus and deuterium MR spectroscopy. Now, there are several challenges to do phosphorus and deuterium spectroscopy, and that has to do with sensitivity. So the sensitivity is determined by the Boltzmann distribution, and for a spin 1/2 nucleus, there's 2 energy levels, and the population of those energy levels is determined by this equation, and there's a delta E in this equation, and that's actually the energy difference between the 2 energy levels. And you can see that this delta E is determined by the gamma, and we have seen for deuterium, but this is also the case for phosphorus, that the gamma is way lower than for proton, meaning that this distribution over the energy levels is much less favorable for X nuclei as compared to proton. So for phosphorus, if we look at a situation at a field strength of 1.5 Tesla, then this ratio between the population of the lower and the higher energy level is this number, which is really close to 1. So that means that the net magnetization is very, very, very small. Only 4 out of 2 million spins. There are only 4 out of 2 million spins in excess in the lower energy level as compared to the higher energy level. And this determines the size of the net magnetization. So if you look at this equation, then an obvious way to increase this number is to go to higher fields. So if you go to 3, 7, 14 Tesla, you see this ratio, at least the last part of this number, you see that increase linearly with the B0. And actually, you get even higher sensitivity gains when the sample is not noise dominant. So for deuterium, we actually looked at the sensitivity dependence on the magnetic field strength, and we did that together with the Kiel group. And this is data in the post-mortem red brain, so the SNR is a function of field strength. You can see that it increases actually supralinearly, and we did the same thing in the human brain. So this is data at 4 Tesla Yale and 7 Tesla in Utrecht with a very similar coil setup. And this is just data at natural abundance, and you can see here these spectra are at the same noise level, and you see a much higher signal at 7 Tesla as compared to 4 Tesla. So we determined that the sensitivity scales with B0 to the power 1.65. And that means that at 7 Tesla, you have 4 times as much signal as compared to 3 Tesla. And you can use that to go to make your scan faster, but you can also use it to go to a higher spatial resolution. So here you see again a comparison at 4 Tesla on the left and 7 Tesla on the right. The top row is with a resolution of 8 milliliter per voxel. And on the bottom, you see a scan with much smaller voxels of 1 milliliter. And we still can see the peaks at 7 Tesla, but at 4 Tesla, it's not really clear what you see in the spectra. So the solution to combat this low intrinsic sensitivity of X nuclei is to go to ultra-high fields. And of course, there's another advantage, and that is the increased spectral resolution at high fields. You saw it already a bit on the previous slide. So if you compare here this 7 Tesla spectrum with the 4 Tesla spectrum, you see there's a better separation between the peaks. But for phosphorus, this may be not even more important. Here you see the same phosphorus spectrum from the liver at 7 Tesla that I showed before. And this is a 3 Tesla spectrum from the liver from the literature. And you see that especially here in this region with the phosphomonoester and the phosphodiester signals in which we are interested, you see much better spectral resolutions compared to the 3T data. Another challenge is the coils. I don't know how many of you do ex-nuclei work, but usually, at least outside the brain, there's only local transmit coils available, so simple surface coils. And they have a limited penetration depth. So if you would have a deuterium or a phosphorus surface coil and you're interested in metastasis in the liver, then with this coil setup, you would only detect this metastasis over here and not that one. Now, if we look at the frequencies of the different nuclei at different field strengths, then we see that actually for phosphorus at 7 Tesla, the frequency is very close to the proton frequency at 3 Tesla. And for deuterium, it's very close to the proton frequency at 1 Tesla. And at those lower clinical field strength, there is really a lot of experience and expertise in RF coil engineering. And so what we did is, okay, we make use of that established RF coil engineering, and we design a coil for ex-nuclei at 7 Tesla. So at 1.5 Tesla, 3 Tesla, usually there is a so-called body coil, which is incorporated into the scanner. And this is a body coil which is used for to transmit at the proton frequency for most of the applications. Now at 7 Tesla, there's not such a proton body coil, and that's because it doesn't work at this high frequency due to the short wavelengths. However, the space is still there. And if a body coil for proton at 3T is possible, then it should be possible to make a body coil for phosphorus at 7 Tesla. Actually, we could more or less take out the body coil from a 3 Tesla scanner and put it in a 7T and use it for phosphorus. So that's actually what we did. Well, we didn't take a 3T coil, but we made a new one. And we actually dual tuned it for both deuterium and phosphorus. And we incorporated that into our scanner. So this is the patient tube that you see here, and the coil is made around it. So on the outside of the tube, what you see here is actually mostly the shield, but this is the body coil. And we put it in the scanner. And then we also made a special receive coil for different applications. So in this case, what you see here is a body receive array, an 8-channel body receive array, which is again dual tuned for both deuterium and for phosphorus. So with this setup, we can detect, transmit and detect deuterium and phosphorus signals throughout the body. And Ayhan Gürsan, he characterized this body coil. So this is the simulation of, or a picture of the coil. And he simulated B1 plus maps. You see here the simulated B1 plus map for deuterium. But we also measured the B1 plus maps for deuterium and for phosphorus in a body size phantom. So here you see the maps, and you can see that the B1 plus field is really homogeneous for deuterium with a variation of only 12%. And for phosphorus, it's a little bit more, there's a little bit more variation of around 19%, but that is very similar to what you will have for a proton body coil at 3 Tesla. And with this, we can collect deuterium and phosphorus MRSI data throughout the whole abdomen and thorax. As you can see here, so on the left is the deuterium data, on the right the phosphorus. And what is really nice is that we don't need to change coils, reposition the subject or anything. So you can make the 2 scans directly after each other with exactly the same planning. So we did it here with the same field of view, the same resolution. So for each voxel, we have both then a deuterium spectrum and a phosphorus spectrum that you can directly compare. So here you see an example of a deuterium spectrum. This was a bit late after administration, so 90 minutes after administration, but you still see the glucose peak here in the liver. And this is then the phosphorus spectrum from the same voxel yeah, similar to the other phosphorus spectra that I've already shown. So this was the situation that we wanted to move away from. Now we have a body coil for transmit in combination with the body receive array, and with this we can assess yeah, all the organs in the body. So then finally, in the last few minutes, I will turn to some applications. So Lieke van der Wildenberg, she used this setup first to look at if there's actually any spatial variations in phosphorus metabolites in the liver. So far, nobody had been able to look at this with a simple surface coil, but now we can cover the whole liver, and actually there appeared quite some spatial variations, especially for the phosphomonoesters and the phosphodiester's. So those compounds that are of interest for tumors. And so we saw actually differences between the right lobe of the liver and the left lobe. And if you look at the bottom row, there's the ratio of PME over PDE, so this tumor marker, which is actually higher in the left lobe of the liver as compared to the right lobe. Then if you move to tumors, it's important to know this, right? Because if there's already changes or differences in healthy tissue, then yeah, you need to take that into account. So as a next step, she studied patients with liver metastasis, and they are receiving chemotherapy. They obtain a CT scan before the chemotherapy and 9 weeks after. So that's the time point where the effects of the chemotherapy are being assessed. What we did is we added some phosphorus scans, also at baseline and at 9 weeks, but then also at 2 weeks after treatment, to see if we can already predict whether the chemotherapy is effective at 2 weeks after treatment. And this is an example of one of the patients. We are still in the initial stages of the study. This patient had a metastasis indicated over here. So these are different slices in feet-head direction. We see somewhat elevated PME over PDE, which actually goes beyond the border of the metastasis. But the bad news for this patient is that after 2 weeks of chemotherapy, this PME over PDE ratio actually only increased. And you would have expected the opposite if the treatment was effective. After 9 weeks, it was even worse, and half of the liver was metastasized. And this was also apparent from the CT scan. But in hindsight, we could probably have predicted that already after 2 weeks. And then there might have been a chance to still change to another sort of therapy. In view of time, I will skip this, but we also can now look at other tissues which were not accessible before, like the pancreas. And we also apply phosphorus MR spectroscopy in the heart. But for the last few minutes, I would like to show some deuterium data. So what happens if you drink deuterated glucose? It will enter your stomach and then go into the small intestine, and via the portal vein, it will enter the liver and then go into the peripheral circulation. And actually, we can really nicely follow that with deuterium MR spectroscopic imaging. So here you see deuterium MRI data overlaid on the image, coronal image at baseline. So you just see the deuterated water signal, the natural abundance deuterated water signal here in the liver. So this is the small peak. Directly after intake of the deuterated glucose, you see a strong signal here in this region where the stomach is located. And then a little bit later, we see a sharp glucose signal in the region here where also the portal vein is located. So we think that this is a signal from the portal vein, or at least the veins leading to the portal vein. And then, especially because in the liver, you don't see a glucose signal yet. So you have to wait a bit longer before you see a second signal in the liver coming up, which is from the glucose. So here you see just one voxel from the stomach. At baseline, there's no signal in the stomach. And then after administration of glucose, you see a strong glucose signal, and that decays with time. So every measurement is about 10 minutes. In the portal vein, you see the maximum signal a bit later. And then in the liver, you have to wait for about half an hour more to see the maximum signal. And we also determined the deuterium enrichment in plasma glucose. And there we see actually the peak right after where we see the peak in the liver, which makes sense because it first has to go through the liver and then it goes to the peripheral circulation. So in this way, we can really nicely analyze what happens with glucose from the stomach all the way to the liver and then to the periphery, which also makes it possible to look at disturbances, for example, in gastric emptying in relation to disturbances in liver glucose metabolism. Okay. This. And then Li Shen, she did a similar study as Lieke, so she also looked at patients with liver metastasis, but these patients were treated with radioembolization. And in this study, we then really combined the deuterium and the phosphorus MRI. So this is the protocol. Patients drink the glucose. Then after 20 minutes, we position them in the magnet. We do some preparation, shimming, etc. Then we start the deuterium MRI scan, and after that, we start the phosphorus MRI without any repositioning and with exactly the same planning. And here you see data from one patient before the start of the treatment. It's two slices, a lower slice here at the bottom where there's normal appearing liver tissue. So you see here the deuterium spectrum in the blue voxel and here the phosphorus spectrum. So in the deuterium spectrum, you see the deuterated water and the glucose, and this is the phosphorus spectrum. But then if we go a few slices higher, where there's the tumor, it's quite different. So especially for the deuterium data, we see a lower glucose signal and a clear signal from lactate, and that we don't see in healthy brain tissue, liver tissue, sorry. And the phosphorus spectrum is actually not that different from the blue voxel, but there's a slightly increased phosphomonoester signal. Then after treatment, so that's 4 weeks later, yeah, it's quite striking. In the normal appearing liver tissue, there's hardly any change. But in the tumor, we see no lactate anymore. And in the phosphorus spectrum, there's hardly any signal left, indicating that there's necrosis, and that's also apparent actually from the PET scan here in the corner, that this tumor has become necrotic, which means effective therapy. I also have some brain data from Narges, but I will skip that in view of time. So we also do brain measurements with exactly the same body coil, but with a dedicated receive coil for the brain. And yeah, it's a simple sequence, the FID MRSI sequence, but it's also slow, which limits our spatial temporal resolution. So we also do some work to accelerate the scans. And this is an example from Kiel where he implemented deuterium APSI to accelerate the scans. So in conclusion, because metabolic changes often precede changes in morphology or function, metabolic MRI can be a very powerful tool for disease staging and especially monitoring early treatment response. Because of the low intrinsic sensitivity, X-nuclei MR greatly benefits from the increased sensitivity at ultra-high field. And with a Bohr coil for transmitting at those X-nuclei frequencies, we can have coverage of all body organs. And the combination of deuterium and phosphorus MSI seems to give complementary information which can be used for early treatment response assessment. So with this, I would like to end, and sorry that I'm a bit over time, but I'm open to any questions.
Thank you very much. You're perfectly in time, actually, and thank you very much for this great presentation, giving you insights into your work. I think there are a couple of questions. So who asks first? Served first.
Should I stop sharing or?
No, please leave it on.
Okay.
So there is a
Can I look at, yeah, I look in the chat. Can I just,
yes,
I can read them and yeah. Okay, so there's a question from Andre. Oh, now the thunderstorms are starting here. Okay. Why does the T1 of lactate for some compounds increase with increasing magnetic field strength? Maybe I should go back to the slide with the T1s if that's possible. Let's see. That's going to be. I think for most of the deuterated compounds, the T1 is actually quite insensitive to the magnetic field strength. Quite a way, sorry. So I'm not sure. And for lactate, I think there was not that much data in the graphs. Never a good idea. Sorry. Yeah. Okay, finally. Yeah, so for lactate, there's actually only one measurement at 11.7 Tesla. So at this time, I actually don't know. So for water and for glucose, the T1 is pretty constant and not dependent on the field strength. For GLX, it seems to go up slightly, but yeah. I think in general, you can say that the T1s are quite insensitive to the magnetic field strength. And that's because deuterium is a quadrupole. For proton, you would see different things, right? Where the T1 increases with the magnetic field strength. I hope that answers your question. I missed the assignment of the signal to lactate rather than alanine, but assume that this is known. Oh. Seems my question is just being answered.
Yeah, I think that Andre answered the questions, at least one of them maybe below.
Oh, okay.
He also wrote that sorry, I mistyped. I meant GDX.
Oh, okay. Or GLX or okay. Well, alanine, I don't know if I think deuterated alanine has not been detected, as far as I know. Does MRSI use phase sensitive detection or magnitude mode? So so as spectroscopists, we really don't like magnitude mode because in spectroscopy you have your absorption peak and your dispersion peak, and if you use magnitude, you will mix the two and you will get broader lines. So indeed, we use phase sensitive detection, and we also phase correct our data to really get the absorption mode spectrum to have the lines as narrow as possible.
We have a new question. Can you please explain the importance of PME or PDE metric? Why is it important and what does it reflect?
Okay, I try to explain this, but so the phosphomonoesters, those are the membrane phospholipid precursors. So the phospholipids themselves we cannot see, they give two broad signals. But the PMEs are the precursors of those membrane phospholipids, and the phosphodiester's are breakdown products of those membrane phospholipids. And in other research, for example, in breast cancer, it has been shown that the ratio of precursors with respect to the breakdown products is increased in tumor tissue. And also that this metric, so this ratio of PME over PDE, that this is informative about treatment response. So in breast cancer, they observed that when the treatment is effective, that this ratio goes down. So that's why it is of interest in cancer research.
And we have a general question from Sanjay Tiwari. Can recycling of lactate into neighboring tumor cells underestimate the amount of pyruvate being converted to lactate, since the recycled lactate will be metabolized back to pyruvate?
Yeah, that's a good point. We just look at the accumulated pool, but that's not probably the net flux of lactate synthesis. And actually, it gets even more complex because after oral administration of the deuterated glucose, it ends up everywhere in the body, right? So also muscles can produce lactate that can then afterwards be transported to the brain and taken up there. So that makes the interpretation actually a bit complicated. So you cannot be totally sure of the origin of the signal, and you can also not be sure if what you detect, whether that's all of the lactate that is being produced or whether it's actually less because it's being recycled. Yeah. There's also a question about slice thickness, but so we don't do any slice selection in our 3D MRSI experiments. So we just do excitation of the whole volume, and we do the phase encoding in 3D in 3 directions. And in the liver measurements, we use isotropic voxel size of 2 by 2 by 2 centimeters, which is still quite big, especially if you go to those patients with liver metastasis. You would like to make the resolution a bit better, which I think sensitivity-wise would be feasible. It's just that it takes then a long time with the 3D phase encoding. So that's where the acceleration techniques are going to be necessary. For example, this FC approach, but you could also think of other approaches like concentric rings or whatever.
There is another question from Andreas Berg. First things first, thank you very much for the didactic presentation. And then can you tell why the ADC in prostate tumor is still reduced after therapy? And then there's a second question too.
So that is it was not my data, and this is also not really my my expertise, but I guess finally in the end you will see those values being normalized, I guess, but this takes a long while. I think the metabolic changes they happen much earlier than really the structural properties of the tissue. So I think this is sort of in general why you would see changes earlier in a metabolic scan than in scans that look at more like tissue morphology. But this is not my field of expertise, so I'm not sure what you would expect for the diffusion-weighted spectroscopy imaging.
Okay, and before Francisco Smolder can ask the question, there's a second question from Andreas Beck. Is there a tissue-dependent impact of quadrupolar fields or ions on T1, possibly influencing signal intensity in MRSI? I think that could be the case. So the plots that are still on the screen, I think that's all brain. So the field is relatively new. So I think this characterization of relaxation time so far has only been done extensively in brain and not in other tissues. So that's indeed something that
for the liver, we should probably look at. We have done some experiments in skeletal muscle, and there you have actually an additional complication because muscle tissue is very structured. So what we see in the muscle is residual quadrupolar couplings in our signals, which makes a bit more complicated. Yeah, so there's definitely specific tissue effects that need to be considered.
Okay.
Yeah. And also what you see here is that the T1s are quite different between the different metabolites, for example, between glucose and lactate. So yeah, so what we typically do is we take an approach with a relatively short TR and a small flip angle, but then yeah, you have to take into account that you're saturating the lactate more than the glucose, for example. Yeah.
Okay. Franciscus, do you want to ask a question?
Yeah, thanks. Very nice presentation, Jeanine. I'm not from this community, so my question might be very simple. If I understand your imaging technique correctly with a phase encoding in 3 dimensions, then this means that chemical shift evolution takes place during the gradient. So isn't there a problem at some point that the delays to use the gradients means that the chemical shift evolution goes beyond 2 pi, and that leads to some form of aliasing? And the other question is, why, in this case, do you get very large linear phase corrections that you need to apply?
Yeah, let's start with the second one. Indeed, there's a linear phase correction that we need to apply, but that's a known linear phase correction. It makes the spectra look a bit less pretty because you get this baseline roll due to the linear phase correction.
Right.
But actually, for quantification of our data, we use the time domain data. So then this delay, it's an acquisition delay, is not a problem at all. It's only for the visualization of the spectra, which they are then a bit less pretty, but otherwise no problem. Of course, there's also T2 relaxation in this delay that you need for your phase encoding gradients, and the T2s are quite short, so you would like to keep it as short as possible, but it doesn't lead to big problems. The thing is that so this delay is exactly the same for each acquisition. So you do multiple acquisitions where you change the amplitude of your phase encoding gradient, but this delay is always the same. So that means that this doesn't lead to something which for example, like chemical shift displacement artifacts or anything like that. Because it's a constant delay during which there is Larmor precession. So in the end, you're not affected by that because it's not changing delay.
And this total time is relatively short compared to the difference of the end of the frequencies of your spectrum, in all cases, deuterium, phosphorus, protein.
Yeah.
All right, that's clear. Okay, thanks.
All right, Dr. Prompers, this is Jan Hövenaar. So thank you so much for being with us tonight. And I'm sorry that I couldn't be there in the beginning, but it was a great presentation. I would like to add one question, that is, I like your approach of changing the entire body resonator, but of course that's not so easy. What we do is, and maybe you know that as well, we use this relatively large torso transmitter and then local receiver arrays. However, already with that transmitter, which is much smaller than your body resonator, we get issues with pulse power, like that applying pulses short enough, for example, for refocusing an adequate bandwidth. How do you deal with that? How do you deal with that? Do you have like extra amplifiers?
Yeah, so yeah, that's a very good point, and there is clear advantages and disadvantages of such a big body coil. So obviously the big advantage is that you have coverage of the whole body and also that you have a homogeneous B1 plus, which makes quantification a lot more straightforward. But the disadvantage is indeed that such a big coil is less efficient. So in terms of B1 plus, yeah, you don't get as high values as you would have with the surface coil. We do have separate amplifiers. So actually we have an amplifier for phosphorus, which is an old proton amplifier from a 3T. And that can deliver 2 times 18 kilowatts.
Yeah, that's a lot.
So that's a lot.
Nice. Yeah.
And for deuterium, we have an old amplifier from a proton amplifier from a 1 Tesla machine, which is 10 kilowatts. It's a single channel, so that's already less. And with that, we can achieve for both deuterium and phosphorus a B1 plus of 10 microtesla, which is still low compared to what you would get from a surface coil, of course, depending on the distance. But and that's why we actually take this simple pulse and acquire approach. So no refocusing pulses, just an excitation pulse, and we use quite low flip angles. I didn't put any details on the protocol, but for phosphorus, we use a flip angle of only 12 degrees with a repetition time of 60 milliseconds. And for deuterium, we use a flip angle of 40 degrees with a repetition time of 100 milliseconds. But this is clearly a disadvantage. So if you would like to do any other sort of fancy pulse sequence, maybe just single voxel spectroscopy or some editing for which you need a 90 and 180 degree pulses, then this is really not possible. Or, challenging. Yeah. So it really depends on what you want to do. Yeah.
And do you, I mean, that's maybe a little bit off topic, but how do you deal with regulatory things then if it's not a medical product anymore, right? If you use the custom body coil and custom transmitters?
No, that's true. And so we need an IMDD, so investigational medical device dossier, which is then being judged by in Utrecht by a special department within the hospital that has to approve. So it's a whole lot of documentation that you need to deliver, and you have to perform a lot of tests which need to be documented before you can use it. That has become a lot more complicated during the recent years.
Sure.
And that's then again an advantage of this bore coil, because you can then use it for basically any tissue. The only thing that you need to change is for brain, of course, you need another receive coil as compared to body applications. But for receive coils, it's much easier to get approval because there's not that much
you don't transmit,
risks as compared to the transmit coil. So that's good. So for the bore coil, you need to go to the procedure only once, and then
thank you so much. Very interesting. Ronald and I would like to thank you very, very much, and everyone here participating in the discussion. So with that, best wishes from Kiel. Again, Dr. Prommel, thanks you so much. That was very, very, very interesting. Thanks everyone for joining us tonight.